Everybody, welcome back to another lecture video, and in chapter 20 we're going to be looking at enzymes and then wrapping up talking about vitamins. So let's jump right in. So enzymes are biological catalysts. We learned that catalyzed reactions can go faster, and a catalyst is just something that will, we can say, lowers the activation energy. This is the energy required to get a reaction going. And the way that catalysts usually make the reaction happen faster is they provide an alternative route for the reaction to proceed through that usually has a lower starting energy. And enzymes are really good at doing this, and we'll look at a few ways that this is accomplished. So they are biological catalysts as opposed to just acid catalyzed, or we use hydrogen gas and or like nickel and palladium and platinum as catalysts when we do hydrogenation or reductions, and those would be inorganic catalysts. So they increase the rate of a reaction by changing the way the reaction takes place. The catalyst itself is not changed in the process, and so the enzymes are going to be available to do this job over and over and over, no matter how many reactions they take place in or take part in. And like we said, it lowers the activation energy, and we show this typical energy diagram. I'll come back to this. So enzymes increase the rate of a chemical reaction. Now they again lower the activation energy. Maybe I will jump to my my activation energy diagram. Looks like it's, I guess this was it right in the beginning. So if this is a typical pathway for, let's say, carbon dioxide and water to go through, the reason the activation energy might be so high has to do with the fact that these have to collide in the right way with the right energy, again in the right orientation, so that they can form bicarbonate and hydrogen ions. With an enzyme, though, we see that the activation energy hump is much lower. And the reason for this is that an enzyme actually will hold the molecules near each other in the right orientation, and actually eliminate some of that randomness of them needing to collide in the right way. An enzyme just pushes them together in the right way. So it's a way of, again, changing the method in which that they react together. It doesn't change the reaction, though. Okay, so we still get the same products. We still need the same reactants. Now, this example, carbonic anhydrase, this is an enzyme in the blood, and it catalyzes, the rapid conversion of carbon dioxide and water into bicarbonate and hydrogen ion. And the reverse reaction is also catalyzed by this particular enzyme. This is important for our blood pH to maintain, a non-acidic environment in our blood. No, our blood actually moves a lot of carbon dioxide around. It's the waste product, and that dissolves in water, and it can form bicarbonate. And this is an important buffer system in our blood. So being able to modulate how fast or slow this reaction happens is how we can change the pH of our blood, and to compensate for, let's say, a more acidic situation, like when you're being very overexerting, right? If you're being very active, you start to build up a more acidic environment in your muscles due to that activity. And so your pH of your blood needs to be able to compensate. Nearly all enzymes are globular proteins, and they have unique three-dimensional shapes that will recognize and bind a small group of reacting molecules called substrates. So every enzyme has substrates, and the substrates are the molecules that it interacts with. So in the previous example, carbonic anhydrase reacts with the substrates carbon dioxide and water in this direction. In the backwards reaction direction, these guys would be the substrates. So the substrate is what reacts. Now, these enzymes have a tertiary structure that includes a region called the active site. And this is where one or more small groups of substrates can actually bind to create the chemical reaction. They have specific amino acid residues in that active site that interact with the functional groups of the substrate. And this helps to form hydrogen bonds, salt bridges, hydrophobic interactions. And so an enzyme like lactase, shown here, so this is the big molecule lactase, you can see it's actually got a quaternary structure. It's got 4 subunits, all the different colors. And you can see that there's an active site here, and it's kind of zoomed in here, where we have the substrate lactose in gray, so lactose here being the disaccharide. It's actually held in place in the active site by hydrogen bonds with the amino acid R groups. So this amino acid, all these amino acids that are present in the active site of this enzyme hold the substrate in place. And remember, this lactose has lots of OHs on it, right, because it's a sugar. So all of those hydrogen bonds are being formed between tryptophan, tyrosine, what is this, glutamine? No, I'm forgetting it. This is glutamine, this is histidine, asparagine, it's not glycine, glutamic acid. No. Glutamic acid or glutamine? Either way, sorry. There are going to be polar amino acids, those that are actually going to interact with a polar substance, such as a sugar. Now, we can classify enzymes a few different ways, and we do that by their specificity. Now, some enzymes show something called absolute specificity. They catalyze only one reaction for a very specific substrate. An example of that would be urease. This catalyzes only the hydrolysis of urea. So an absolute specificity means one substrate only. Now there can be some enzymes that can catalyze a reaction of 2 or more substrates. We typically call these either group enzymes or linkage type enzymes. And so they can be very specific for a group. So for example, hexokinases add phosphate groups to any hexose sugar. So that just means it has to have 6 carbons. And any 6-carbon sugar can get a phosphate group added to it by a hexokinase. You can also have enzymes that recognize certain linkages. So here you can see chymotrypsin, for example. It'll catalyze peptide bonds. And there's a couple peptide bonds that will actually hydrolyze, and we'll look at that a little bit later. So if it's the bond type that it recognizes, then it's linkage. If it's particular groups, then it would be group. And if there's only one substrate, then we would call that absolute specificity. So group specificity, linkage specificity. And so 3 general sort of substrate type of enzymes. Now, the combination of an enzyme and a substrate forms something called the enzyme-substrate complex. You can kind of see it here. So here we have a substrate, here we have an enzyme, and this, in this case, lactose, has the right shape to fit into the active site of the enzyme lactase. So E, representing enzyme, and S, representing substrate, form together in an ES complex. This ES complex is that alternative pathway with the lower activation energy. So what that means is the reaction happens faster. And again, what holds this substrate into the active site is those interactions with the amino acids that are in that active site of the enzyme. So the same interactions that hold proteins together hold molecules in the active sites. Now, when our enzyme has done its job, it will have converted from an ES complex into an EP complex, an enzyme product complex. So here, the important bit to know is that the ES complex is about to catalyze. So notice that this bond here, this ester bond, hasn't been broken yet. Well, that's not an ester bond. This anhydride, or this what is it? This acetal bond. And here it has been broken. And so here we have 2 monosaccharides. And so the last thing that happens is that this enzyme product complex needs to dissociate. So it'll give us our products back, and then we have an enzyme that's ready to go back into circulation. It also helps that the active site can be flexible. So here in this example, you see sort of a flexible active site in lactase. And the flexible substrate lactose will adjust to actually provide the best fit for this hydrolysis reaction. And then once it gets hydrolyzed, it gets released. And so that's one of the ways that substrates can kind of interact with an active site. Now, that wouldn't be the lock and key model, what I just described. That would be more of the induced fit model, where them being flexible, that the active site and the substrate, actually allows for some adjusting and some, best fitting sort of going on. They kind of help change their shapes a little bit to best fit. This also explains the group specificity, because having an active site that can adjust a little bit and having substrates that can flex or bend a little bit means you have a wider range of things that can actually fit into that active site and be catalyzed. Whereas the lock and key model is very rigid. This would be kind of explaining the absolute specificity, where only one substrate actually fits in there perfectly to allow the reaction to happen, or in this case, like a key fitting a lock, right? Every key doesn't open every lock. It needs to be a very specific key shape. So the induced fit model has the substrate and enzyme working together to acquire the best fit and the best orientation, and this is what actually adds to that lowering of the activation energy, them being able to be brought in as close together as possible for this reaction to take place. So there's two models of enzyme action, lock and key, and then induced fit. So quick learning check. So pause this, see if you can get the right answers here. So which is the active site? It is the section, a section of the enzyme. It's not the entire enzyme, it's definitely not the substrate. In the induced fit model, what happens to the shape of the enzyme? It adapts. In lock and key, it would stay the same. All right, an enzyme name does describe its action. Sometimes it can even give us information about what kind of substrate it reacts with. So in this part of the chapter, we're going to look at classification of enzymes, and we're going to give some names, or look at how we give enzymes their names. I'm showing you here the ribbon structure for alanine transaminase. Kind of tells you what it does here. This is an aminotransferase, and you can kind of transaminase means that it's going to move an amino, and this one looks like it does it on an alanine. So this one actually contains 495 amino acid residues. So that's a pretty big protein. I guess this gray thing here would be the alanine. All right, let's look at a few examples. So when we're naming enzymes, the names usually end in ASE. They identify, the name can identify the reacting substance. So sucrase is a reaction of sucrose. We have amylase in our saliva. This helps break down amylose, just as an example. The name can describe the function. For example, it can oxidases, right, catalyze oxidations. So there's a lot of different names. There's the specific name, there's the category name, there's the type of substrate specificity, right? So there's a lot of things that we can say about proteins or enzymes. So don't let that confuse you. And it can also be a common name. So for example, pepsin and trypsin are just names of enzymes that we talk about or that we would use all the time. These happen to be peptide bond hydrolyzers, so they can go in there and actually cut. So these would be kind of linkage enzymes, right? They go and they cut peptide bonds. Now they'll cut different ones, and we'll look at that a little bit. But their name doesn't really tell us much, right? It's just a common name. So we can have names that are specific, we can have common names, we can also have sort of classification names like oxidases. So let's go through a few examples of enzyme classes. So the first enzyme class is the oxidoreductases. So these are involved in oxidation and reduction reactions, just like it sort of implies by the name. Oxidases would catalyze oxidations. Let's see. We don't call the reduction ones oxidases, and I think something that does a reduction reaction, but it might get a name such as dehydrogenase, right? And actually, a dehydrogenase would actually be an oxidizer as well. But just two examples of enzymes that would fall under oxidoreductases. So oxidases in general, and then a specific one, dehydrogenase. And this one removes, actually it looks like it can also add two hydrogens. So in that case, a dehydrogenase might also be involved in reduction reactions. So, some of them can catalyze reactions both forward and backwards. This one in particular, alcohol dehydrogenase, looks like it only catalyzes the removal of some hydrogens. So it actually removes some hydrogens from ethanol to make ethanal. So there's an example of a oxidoreductase. In this case, this is oxidizing alcohol to aldehyde. We have transferases. Transferases catalyze the transfer of a functional group. So transaminases, like we talked about right at the beginning, that move amino groups from one molecule to another. Or kinases. Kinases transfer phosphate groups from one substance to another. So moving a group from one molecule to another is a transferase. Here you can see alanine getting transaminased here. So alanine gets its amino group removed. Alanine then becomes pyruvate. Here we have alpha ketoglutarate that is going to gain that amino group, and alpha ketoglutarate becomes glutamate. So alanine transaminase is responsible for making this reaction happen faster, but also the name here tells us a lot. It works on alanine, and it's transaminase, meaning trans amino, moving an amino group. We have hydrolases. Hydrolases use water to cut a compound into two. So hydrolases like proteases, which hydrolyze peptide bonds, lipases, which hydrolyze ester bonds, or nucleases, which hydrolyze a phosphate ester bond that we haven't looked at yet in the DNA backbone. But all of these, and we can even talk here about the sugar, let's see, what would we call them? Carbohydrate aces? No, they don't call them that. But if I knew the classification name for all of the different enzymes that break down carbohydrates, we could probably include them here as well. Of course, we know amylase, but that just happens to hydrolyze. So I guess amylase would be a hydrolase. Amylase, which breaks down starch, hydrolyzes that acetal bond. So that would work as well. It'd be adding water. It would be cutting. Amylase would be a hydrolase. You can kind of see that here. This is a polypeptide, so we've got NCC, right? NCC, two amino acids here. In this case, a protease comes in and just splits that into two. So we would cut off one amino acid residue at a time. Now we'll talk about chymotrypsin and trypsin, and I might give you a little more info on which specific amino acids those tend to look for, but those would be trypsin and chymotrypsin and pepsin would all fall under these proteases. So they would be hydrolases. We've got liases. This one sounds very similar, This catalyzes the addition or removal of a group without hydrolysis. So in this one, it's not quite the same as splitting a molecule into two. This one isn't also the same as transferring a group from one molecule to another. This one has adding a group or removing a group without hydrolysis, so no water involved, or at least no water used in cutting. You can see here that there are some examples that have water involved. So a decarboxylase removes CO2, a deaminase removes NH3, dehydratases can remove water, hydratases can add water, carboxylases can add CO2, aminases can add NH3, and so on. This example, pyruvate decarboxylase, has a pyruvate molecule losing a CO2 to form ethanol and a carbon dioxide. So decarboxylase here, removal of a group. We have isomerases. Isomerases rearrange molecules, or isomerize them. So here it's just rearranging, nothing added new. So isomerases can rearrange reactions. Epimerases can actually rearrange carbohydrates. So if there's a enzyme involved in turning fructose into glucose, it would be an epimerase, because remember, the sugars are epimers of each other when they're not mirror images. We called them enantiomers when they were mirror images of each other, but they're all epimers of each other, the different sugars with the different L and Ds when we flip those. Okay, so isomerizations can be done by isomerases. Last but not least, we have ligases, and this is joining two molecules together. This usually involves ATP, and so synthetases and carboxylases that use ATP would fall under this category. So if we're forming a bond between carbon dioxide and a substrate and it uses ATP, or if we're forming the bond between two substrates and we're using ATP, these would be ligases. Pyruvate, carbon dioxide, and ATP energy produces oxaloacetate. ATP, having lost one of its phosphates, is now ADP, adenosine diphosphate, and that phosphate group gets liberated. So this is using ATP for energy. This is what happens after we've freed up that bond that produced enough energy to make this reaction happen, to add the CO2 to this molecule. And then something has to come back and remake an ATP out of this, and again, that costs energy. All right, so enzyme classes. So if I give you an example on an exam of a reaction type, your job would be to try to classify it according to these examples. Let's try it. So go ahead and pause the video, give these a shot. So if we're converting a cis fatty acid to a trans fatty acid, I would hope you'd say isomerase. If we're removing some hydrogen atoms to form a double bond, I hope you would call that a dehydrogenase, and that would be an oxidoreductase. Combines two molecules to make a new compound. A synthetase would do that. Adding NH3, this would be an aminase, at least from these four categories that you're picking from. All right, now if we were adding two molecules together, this could also be a ligase, right? If it involves ATP, just to kind of throw that out there. Okay. Yeah, I don't think there's any others here that we can kind of like overlap names with. But yeah, number C here. If it had ATP involved, that would be a ligase, right? More specifically. Let's talk about enzyme activity. The activity of an enzyme actually describes how fast an enzyme catalyzed reaction can go. And enzyme activity is strongly affected by certain conditions, such as temp, pH, concentration, not your concentrating, but concentration of both enzyme and substrate. It matters how much enzyme we have, matters how much substrate we have, and what kind of um, environment in terms of temperature and pH. So let's go through some of the factors that make enzymes work better, work less, and so on. So temperature is probably one of the easiest to understand. Remember, enzymes are tertiary or quaternary globular proteins, so they're all held together by different interactions of the R groups. Now, what happens is that if we increase the temperature and we look at an enzyme's activity, we can see that at low temp and at high temp, there's not a whole lot of activity, and for different reasons. At low temp, there's not a lot of motion. So molecules don't move around a lot, so they don't come into contact as often, so we don't see as many enzyme substrate complexes forming. At high temperature, denaturation happens. The energy inside the molecule gets so high that it actually overcomes the hydrogen bonds and salt bridges and all the things that would hold that structure together, folded as a tertiary or a quaternary enzyme. So if those bonds fall apart, then the molecule can't stay together and it definitely can't do its job. So there is an optimum temperature range, and usually for our bodies it's about 37 degrees, where as long as you have an enzyme that isn't denatured, that's sufficient energy for it to kind of be doing its maximum work. If you increase the temperature, there will be a slight increase in the reaction rate, but more likely than not, the enzyme will be too misfolded to actually do its job. And so depending on the organism, this temperature can be slightly different. For us, 37. There are organisms called thermophiles that can actually live anywhere from 50 degrees Celsius to 120 degrees Celsius. I mean, that's really, really hot. And their enzymes and their tertiary structures aren't destroyed by these high temperatures. We actually use a enzyme, I think it's an enzyme, actually it's a molecule called Taq polymerase. We use it in a process called PCR, where we take DNA and we heat it up so that it kind of splits open, and then we have an enzyme that runs in there and makes copies. And every time we heat up the solution, the DNA splits, it makes copies, and then we cool it, and they all like condense into new DNA strands, and then we heat it up again, and those split, and we make new copies. And the reason that this works is because heating up the temperature would normally break down all the enzymes that do DNA copying, but because we've stolen that enzyme from organisms that could live in really hot water, those enzymes work at high temp, and so we're able to do this. We'll talk about PCR more when we get to that chapter, the DNA chapter. But anyways, temperature has an effect on enzymes. Now, pH also has an effect on enzyme, and for similar reasons. You remember that your tertiary structure is held together, right? Your protein is held together by salt bridges, hydrogen bonds, those types of interactions. Well, if we get outside of a pH range, you'll remember that your amino acids can lose their charges. The R groups can actually become positively or negatively charged or lose those charges altogether if the pH changes, as well as the ends of our molecule. We normally say that the nitrogen is positively charged and the carbon is got that negative oxygen on it. But if the pH changes too much, there's an H here instead of a negative charge, or there's only two H's here instead of a positive charge. And again, that can happen on the side groups as well, and the R groups. And different pHs are going to cause these groups and the R groups to have different amounts of hydrogen, essentially. And their charges may appear or disappear. If charges disappear, molecules or proteins aren't going to have those associations that were holding them together. If they appear when they weren't supposed to be there, now they're going to add new repulsions or new attractions and cause the protein to possibly fold differently or to unfold altogether. And so every enzyme has sort of a pH range in which it is working its best. And as you start to deviate from that range, we get subtle changes to the structure of these amino acids that ultimately lead to the whole protein falling apart or not staying folded. And so again, similar to how the temperature can cause this protein to fall apart, the pH is also very important. Too low or too high changes those charges, changes our interactions. So for our bodies, a pH slightly above 7 is that optimum range. You can see here there's actually a different range for different proteins, different enzymes. I probably should have said that. In the stomach, where an enzyme is going to be working in the stomach, you would expect the pH to be really low for it to be in its most productive and active form. If something is active in our blood, then it's probably going to have normal blood pH, around 7.2. If it's active in an acidic or in a basic part of our bodies, it's going to have an optimum efficiency in that pH range. And this is also how we can control enzymes. For example, pepsin is active when it's got its pH 1.5 to 2, meaning when it's in the stomach. But pepsin's not made in the stomach. It gets made somewhere else. And so before it gets to the stomach, we don't want it necessarily doing the job of breaking down peptide bonds. And so it's convenient that when it's not in the stomach, it's also not in its optimum pH, and therefore it's inactive. And so until it gets into its optimum pH, you'll see that there are certain enzymes that are just going to be inactive so long as they're in the wrong pH condition. And that's a way of your body being able to monitor or control, I should say, when these enzymes are active and have more control over when they're doing their job during production and when they're being moved to their proper spot in the body. Trypsin, lipase, arginase, you can see all of these have slight different pH ranges, and we would determine these experimentally. We could use pepsin as a baseline and sort of put it into different pH conditions and see whether it performs better or worse, and that's kind of how we build these ranges. Now, enzyme concentration also has an effect on how well that enzyme activity is, and enzyme activity, of course, we can just look at how fast the reaction goes or proceeds by. So reaction rate. If we increase the enzyme concentration, it makes sense that the reaction rate should go up. More enzyme means more substrate is available to be able to bind. Now, assuming that we don't run out of substrate, this is going to go up, up, up. Now, if there is a situation, right, where substrate concentration might eventually run out, which would cause a flattening of the curve. Or I guess an overall stopping of the reaction. That makes more sense. If we run out of substrate, of course the reaction stops. So long as there's substrate, more enzyme will always lead to a faster rate. Now, as far as substrate goes, increasing the substrate doesn't always mean faster rate. Initially it does. Think about the situation where you have nothing but enzyme and no substrate. So of course your reaction rate is 0. As you start to add substrate, you start to see more reaction happening, and so the rate goes up. You're going to hit a saturation point though. At some point you're going to have so much substrate that at any moment every single enzyme molecule is busy. And when that happens, we see this flattening out of the curve here. This shows that we're at our maximum enzyme activity, and this is because all enzyme molecules are busy forming substrate complexes. And so no matter how much more substrate we add, they all just have to wait their turn for an available enzyme. And so activity reaches a maximum and then stops. And so again, we can look at the substrate activity or a curve like this and kind of see, well, I guess we wouldn't ever make the curve to look at it, but we can see here that substrate concentration has a limiting effect on rate, or a limited effect. It does increase rate up to a point, but then we hit, maximum rate because all of the enzyme is busy. Now we could add more enzyme, and then this would go up again, similar to this curve. So as long as there's more and more enzyme, you can keep going faster and faster and faster. So substrate concentration does have an effect on the maximum rate. Okay, quick learning check. So sucrase has an optimum temperature of 37 and an optimum pH of 6.2. Determine the effect of the following on each of the rate of reaction. So for A, B, and C, decide whether there'd be no change, an increase, or a decrease. So go ahead and pause it, give that a shot. So increasing the concentration of sucrase should have an effect on the rate, right? Up until a point. It won't indefinitely or infinitely raise the rate, but it will get us to a maximum. Changing the pH to 4. Well, even if we were changing 6.2 to 9, going outside of the range is probably going to have a decreasing effect. Anything moving away from the optimum, which is the fastest that it'll be at, is 6.2, so anything other than that is going to be a decrease. Running the reaction at 70 when the optimum is 37. Well, that's probably going to have the effect of decreasing as well. Assuming that 37 is the optimum because higher temperatures start to lead to denaturation, then 70 is clearly going to be a denatured protein or denatured enzyme that's going to have limited enzyme ability. So decrease for sure. Good job if you got all of those right. Let's talk about how enzymes can be regulated. So first off, I'm kind of showing here a type of regulation, phosphorylation, done by kinases. Kinases, in this case, transfer phosphate groups. What would we call this if it was the classifications of enzymes? So this adds a phosphate group. We could call this a transferase because it adds or removes a phosphate from a molecule. The objective here is to look at how enzymes are regulated. So we'll look at allosteric enzymes. We'll talk about feedback control and various covalent modifications. But you can see here, this kinase can actually turn this enzyme on by putting a phosphate group on it, or turn it off by turn it on by adding a phosphate group, or turn it on by removing a phosphate group. So phosphorylation has lots of different effects. So enzyme regulation, the rate of the enzyme catalyzed reaction is controlled by regulatory enzymes that can, for example, increase the rate when more substrate is present. They can decrease the reaction rate when something's not needed. Substance is not needed, right? So think about that. We want more reaction rate when substrate is there. For example, you have enzymes that break down glucose. You don't always have an influx of glucose, so you want those enzymes to kind of chill out when you're not eating, right? And then when you eat some food, you got a lot of glucose, then you want them to kind of kick on. Same thing, when a substance is not needed, say that you're making lots of tyrosine. If you have plenty of tyrosine in your body, you don't want to keep going through the process of making tyrosine. So you need a way to shut off enzymes. And so we're going to look at how enzyme activity can be regulated by what we're calling allosteric enzymes, feedback control, and covalent modifications. So let's look at allosteric regulation first. Allosteric enzymes. Now these bind with a regulator molecule at something called the allosteric site. So the allosteric site is something different from the active site. Allosteric just means other site. So an active site is where the substrate goes, but the allosteric site is different. And when a regulatory molecule, so we'll call this guy here in orange, the regulatory molecule, when it binds to this enzyme, it changes the shape of the active site. So you can see here in positive allosteric regulation, this is a positive allosteric regulator. This comes in here and actually binds with this enzyme to create an active site that better fits the potential substrate. So while this enzyme was looking like this, it wasn't doing its job. So now, to turn it on, this molecule binds to it, the active site changes its shape, and now it recognizes substrate. And that substrate can then go and be converted to product. So this allosteric regulator turned this enzyme on. Now, similarly, we can have negative regulators. So this purple molecule here is a negative regulator. So this one, rather than binding to it and changing the shape of the active site so that it's more likely to react with substrate, this one decreases the likelihood that it'll be able to bind substrate. So it changes the active site to prevent the proper binding. And this, of course, decreases the rate of the reaction because they can't bind, and so they can't form product. So again, this is allosteric enzymes. Allosteric enzymes have an extra site where they can be regulated by allosteric regulators, either positive regulators or negative regulators. Let's talk about feedback control. In feedback control, this is when we have an end product of a reaction that the levels are high. Now when this happens, the end product of a series of reactions, that end product can actually be a negative regulator. So let's take a look at this. So we've got, we'll start here. So we've got enzyme, no, we won't start there. We'll start here. So substrate binds to enzyme. They form enzyme substrate complex 1. They form this square product. That square product then goes and binds with another enzyme. So this is an enzyme substrate complex 2. This is a chain of reactions that's happening. That produces this purple thing, and this purple thing goes and has its own reaction and produces this circle. Let's say this circle is like tyrosine or something you made. Now, we don't want to keep making tyrosine if we have lots of it floating around. And so what would happen is that end product would actually be a negative regulator for the very first enzyme. So now, it being present, right, in that allosteric site, so here we show it kind of binding, that changes the shape of the active site now so that no more S can bind. And so long as there's plenty of this around, no more S will bind, so none of these reactions will happen. And then as soon as the level of this circle product goes down enough so that, you know, most of your enzymes don't have this, you know, negative regulator binding to the allosteric site, then the reaction starts kicking up again, and S starts being turned into green, and green starts being turned into purple, and we get more of these circles. And that's how the end product can actually stop the initial reactions and slow this down. So that's feedback control. And the feedback control ends when the end product is low. When the end product's high, then it blocks that reaction again. Let's talk about covalent modification. So covalent modification involves forming new bonds, covalent bonds. So we can modify activity by covalent bonds. This can either be broken bonds or formed bonds. This is a reversible type of modification or regulation. And zymogens, or proenzymes, are an example of these covalent modified enzymes. They get made inactive so that they can be activated later when they're needed. We'll take a specific look. So here's a couple of zymogens. Now again, zymogens are inactive enzymes that can then be activated. Digestive enzymes, protein hormones, insulin, blood clotting enzymes, these are often formed as zymogens. Again, proteases or digestive enzymes that hydrolyze proteins, again, produced in their inactive form so that they're not doing the job where they're being made. So chymotrypsin, for example, is made in the pancreas. We don't want it to do its job there. We want it to do its job in the small intestine. So then in the small intestine, it's activated into chymotrypsin. I called it chymotrypsin here. I'm sorry, chymotrypsinogen. Pepsinogen gets made in the gastric chief cells, but in the stomach, it gets activated into pepsin. Trypsinogen into trypsin. Fibrinogen into fibrin, right? Proinsulin into insulin. Now, some of these are inactivated so that they can be transferred to the proper part of the body. Some of them are just inactivated so that they can be activated when they are needed in the same part of the body. But again, regulating the enzyme activity. This is an example of, I think this is, what is this? This is trypsin. So when trypsin, the zymogen, is formed, let's see, here, I don't remember the exact example here. So it can be transported to where it's needed. Where's trypsinogen? So pancreas, small intestine. So in the pancreas, oh, but we're looking at chymotrypsin, sorry. Yeah, same thing. So in the pancreas, it looks like this. So this is the inactive form because it still has these little dipeptides that are in the sequence. And so what happens with this one, trypsin actually catalyzes the removal, so trypsin being another peptidase, catalyzes the removal of these groups, allowing chymotrypsin to now be in its active form. And so these trypsinogen and chymotrypsinogen, they're stored in the pancreas until after food is ingested, and then they get released when triggered by hormones from the pancreas, and then they get modified so that they're turned on. So this is just one example of a zymogen, but lots of molecules actually do this. Now, phosphorylation is a type of covalent modification. So you can see here, it's when an inactive enzyme undergoes covalent modification with a phosphate group, and this can either activate it with the addition of the phosphate group, or deactivate it with the removal, or it can activate it with the removal and deactivate it with the addition. So it just sort of depends. And again, we kind of looked at this one already. Both of these are being activated, one by removing the phosphate, one by adding the phosphate. So phosphorylation is just another way of turning on or turning off enzymes. And you can think of this as having a similar effect to allosteric inhibition. Activating or inactivating with phosphate changes the shape of the molecule a little bit, makes it easier or harder for substrate to bind. Quick learning check. So indicate whether the following statement describes an enzyme regulated by allosteric, zymogen, or sort of covalent modification. So hopefully for the end product attaches to the regularity site of the first enzyme. That would be an allosteric enzyme. It would also be feedback regulation. Proinsulin forms in the pancreas. Proinsulin would be a zymogen of insulin. Phosphorylase kinase deactivates pyruvate dehydrogenase. Again, kinase remove or add groups. Phosphorylase kinase here kind of tells us it's a phosphate that's being added or removed, but covalent modification for that one. We can actually use isoenzymes. So isoenzymes are different forms of an enzyme that can catalyze the same reaction, but in different cells or different tissues of the body. They have quaternary structures with slight variations in the amino acid subunits. And so, for example, lactose dehydrogenase, LDH, actually has 5 different isoenzymes that catalyze this reaction of lactate into pyruvate. And the reason why this is useful is that when people have heart attacks, myocardial infarction, it can actually be indicated by an increase in the levels of creatine kinase, which is one of the enzymes, and LDH. LDH. Both of these can indicate that you've had a sort of a myocardial infarction, a heart attack. And you can see here the enzyme CK spikes really early on, and the LDH one sort of spikes again. And different forms of this enzyme actually allow a medical diagnosis of damage or disease to a particular organ or tissue. So, for example, the different isoenzymes of the LDH indicate damage to different parts of the body. And so, when we look at the isoenzymes, if we see, so it comes in two different sort of forms. It comes with an H chain and an M chain. And so we find all H chains, H4, in the heart or kidneys, H3M1 in red blood cells, heart, kidney, brain, and you can see all the way down to 4Ms and no H's, skeletal muscle and liver. So by actually figuring out which one of these enzymes is having the increase in the blood concentration, and knowing which variant it is can tell you which part of the body is actually releasing those enzymes. So it can be used again to kind of indicate where damage is happening in the body. So it's useful. The fact that enzymes can actually have different sort of flavors, right, in different parts of the body. So let's talk about enzyme inhibition now. We've talked about regulation. Let's talk about how we inhibit enzymes. So the goal here is to describe reversible and irreversible inhibition. And the differences are going to also include competitive and non-competitive inhibition. And so I'll come back to this graphic in a minute. So inhibitors are molecules that cause a loss of catalytic activity. They prevent substrates from fitting into the active site, and they can be reversible or irreversible. Now, as far as the reversible ones go, these cause a loss that can be restored. So loss of activity, but can be restored. And that seems obvious. They can act in different ways, but do not form covalent bonds with the enzyme. So that's the key here is no covalent bonds. And sort of that's going to be the irreversible, I can't spell. The irreversible ones are going to be covalent. There's an I there. So the reversible inhibition can actually have two flavors. It can be competitive or non-competitive. So let's take a look at those. The competitive inhibitors, excuse me. The competitive inhibitors have chemical structures and polarities that are similar to the substrate. So they already look like the molecule that the enzyme normally interacts with. So they look and feel like the substrate. So they compete with the substrate for the active site. And so you can see here, we have an active site, there's a substrate. We could form an enzyme substrate complex that would lead to the enzyme product complex and to our product. The trouble is that an inhibitor that has the same sort of features can also bind with our enzyme, and when that happens, it just stops the substrate, for being able to do it. So instead of there being 100 enzymes running around doing the job on the substrate, there might only be 60 enzymes or 50 enzymes that are actually available to interact with substrate because the others are busy interacting with inhibitor. So it lowers the rate by just creating some competition. We like this. Some bacterial infections, right, are treated with competitive inhibitors. We call them antimetabolites. Which one is it? Sulfanilamide competes with p-aminobenzoic acid, which is an essential metabolite for bacteria. So bacteria need this, and so we stick this in their bodies. It interacts with that same enzyme, but stops the reaction from happening as fast. And if this is a metabolite, it means it's important for growth, and so we effectively stop it from being able to grow. Succinate, another molecule that can have an inhibitor in malonate. Malonate has a similar enough structure that it'll interact with that enzyme, but not let succinate interact with that enzyme. So again, we like to use or to take advantage of this competitive inhibitor in medicine. Now, non-competitive inhibitors do not attack the active site. They don't compete with the substrate in that sense. They don't look like the substrate. These have, they just bind to the allosteric sites. So it's similar to allosteric regulation, right? Where we can bind to the allosteric site to change the active site to either make it better or worse, turn it on or turn it off. Here, it's not done by a regulator, though. It's not your body maintaining control over the enzyme. This is just some other molecule that has that same kind of effect. So it's a non-competitive inhibitor because it inhibits by changing the active site, but it does it in that allosteric way. So just binds to another site somewhere. So here we see the inhibitor binding to the enzyme, changing the active site shape so that it can no longer bind substrate. And so we don't get any products from this because the inhibitor it has, changed the shape of the active site. Now, one of the things that you can do when you look at a graph of activity versus substrate concentration is that for a non-competitive inhibitor, adding more adding more substrate has no effect. Because here it doesn't matter how much S there is, I is changing the shape of the active site. So we can't overcome inhibition here by adding more substrate. On the other hand, when it was competitive, adding more substrate actually reverses this inhibition. It creates a situation where there's so much more of this floating around that it outcompetes the inhibitor. So while we can change the rate of a competitive inhibitor by adding more substrate, we cannot change the rate of a non-competitive inhibitor because more substrate doesn't change anything. The inhibitor is going after a different site. Let's talk about irreversible inhibition. So irreversible inhibition happens when an enzyme activity is destroyed because an inhibitor covalently bonds with the R group of the amino acid. So you can see here we have the enzyme serine, or an enzyme with serine. So this is just sticking out of the enzyme substrate, or the enzyme, this is just the functional group of an amino acid in that enzyme. It's just, we're showing that part. This molecule here, DFP, happens to interact with serine. So if there's a serine showing on your enzyme, it'll form a bond here. So now you can see all of this has been added to your enzyme. Where it used to just have an OH, now it has all of this junk. This is going to be an enzyme that can no longer bind substrate. Especially if this happened in the active site. If this is in the active site where we have a serine showing, that serine probably had a job. It probably was there to hold a hydrogen bond of the substrate molecule, or to just be polar so that the polar molecule could like it and stick there. But by changing it, we've changed the shape, we've changed the polarity, things aren't going to fit in there anymore. And so this is irreversible because we formed a covalent bond. We formed a new bond that, again, isn't going to be, we don't have an enzyme to go and break that bond, so this is now permanent. Irreversible inhibitors, here's just a couple. Cyanide, for example, it binds to the iron in your heme, in your blood, so you are no longer able to transport oxygen. It also inhibits your electron transport chain. We'll talk about those things down the road. Just as an example, right? Penicillin is another one. This interacts with the enzyme that helps build the cell wall. So we've got some derivatives of that, different molecules that all seem to work. They all come from the penicillium fungus. These are all irreversible enzyme inhibitors. So to summarize, the shape of the inhibitor, if that shape is similar, it's competitive. If it's not similar to the substrate, it's non-competitive. And it may or may not, let's see, if it's irreversible, it doesn't necessarily have to have a shape that's similar. It just needs to bond with a covalent bond. Binding to the enzyme, competitive binds at the active site, whereas non-competitive binds somewhere else. And if it's irreversible, it forms a covalent bond with that enzyme. As far as reversibility goes, adding more substrate does does not. No, it does. It does increase the rate for competitive inhibitors. More substrate just means more competition. For a non-competitive inhibitor, adding more substrate does nothing. And in terms of reversibility, if it is permanent, then it is not reversible. So this is just sort of summarizing the inhibitors, what they do when they're competitive, non-competitive, and irreversible. So quick learning check. Go ahead and pause these, pause it and give these a shot here, identifying as either competitive or non-competitive. So increasing substrate reverses inhibition. That's competitive. Binding to the enzyme surface but not to the active site, non-competitive. Structure similar to that of the substrate, competitive. Inhibition not reversed by adding more substrate, non-competitive. So I hope that's not confusing. Spend some time and think about these, get them right, so that you're not second guessing yourself on the exam. Let's talk about enzyme cofactors, and then we'll transition into vitamins. So cofactors, let's talk about them. This is just a picture showing some vitamins that you can get from citrus here. We'll talk about vitamin C in a minute. So we have a couple definitions here of enzymes. A simple enzyme doesn't need to have any cofactors. A simple enzyme is just a protein, and it can interact with a substrate. Now, many enzymes, though, are only active when they have an extra part, and this could either be a cofactor or a coenzyme, and there's a lot of overlap with these names. The cofactor is a small molecule or a metal ion, and so typical cofactors are shown here. Copper ions, iron ions, zinc, magnesium, manganese, nickel, those kind of things. A coenzyme is much larger. It's usually an organic molecule, and a vitamin typically fits the bill. So coenzymes tend to be vitamins, cofactors tend to be small metal ions. You can kind of see here how it works together. So here, carboxypeptidase, for example, requires a zinc 2+ cofactor. This is for the hydrolysis of the peptide bond of a C-terminal aromatic amino acid. So this thing always cuts the end off of a protein chain. And so this would be our protein chain right here. It looks like it's always looking for, this one looks for aromatic amino acids. And when it finds an aromatic amino acid, you can see the aromatic amino acid sort of fits into this hydrophobic pocket. And then this cuts the C-terminal side of that. And so the only way this works is if the zinc is here, so that it helps orient the molecule in the right way so that hydrolysis can happen. So it's really neat the way that enzymes utilize different ions and different cofactors and coenzymes to make these reactions happen. It's even more interesting when you actually look in those active sites and see the way that the amino acids are all doing a very specific job there to help get that molecule, the substrate molecule, into the right orientation. Functions of cofactors. They are needed so that enzymes can become active. So for example, you can see here this active site doesn't have, or it has a lot of room in here, but maybe this cofactor is required because it has something on it that'll help the substrate fit in there better. Just like we saw in this previous example, without the zinc, that molecule of the peptide's not going to orient the right way. So similar to that, right? They help the active site. You can kind of say that the cofactors help the enzymes become active. That makes the most sense. And then once they're able to release those products, that cofactor then can be recycled. If it's a cofactor that gets used up, then we need to have it in our diets. We need to have lots of it. Okay, so vitamins. These are organic molecules that are essential for growth. They are required in trace amounts because they're often cofactors, or coenzymes. Some need to be obtained from the diet, and so we'll take a look at which ones those are. And we can classify them as being water-soluble or fat-soluble. So that's the real classification of the vitamin types here. So let's go through the water-soluble ones first. These are the ones that we eat. These are the ones that we can't store in our body because they're soluble. That means we pee them out, essentially. The ones that are fat soluble can be stored in our bodies, and so those are the ones that we don't have to, let's say, get from our food every day. There are many are cofactors. Again, we excrete them, so we can't hold on to them. And these are easily destroyed by heat, oxygen, ultraviolet light. And so when you make your food, the way that you prepare it can often impact how many vitamins actually make it into your body. So I'm not versed on that 100%. I don't know if freezing or microwaved food or, I feel like steaming your veggies is the best way to preserve those vitamins. But again, heating them too much can actually break the molecules apart. So even though you're getting all the atoms, they're not in the right form to be a vitamin. All right, so let's take a look at the different vitamins. So I'm not going to test you guys on this stuff. So really, this is just for your information. So let's just kind of go through it quickly. You can read this more detail if you're interested. So obviously, thiamine, vitamin B1, riboflavin, vitamin B2, um, niacin, vitamin B3. Looks like all of these so far have so thiamine looks like it has a function with its aldehyde group. So it has an important role with that. Riboflavin and niacin look like they're important for electron transfer. Um, Coenzyme NAD+, we'll talk about that one in metabolism a little bit later. Pantothenic acid, or vitamin B5, looks like it has acetyl group functions. Pyridoxine B6 has amino groups. Folic acid B9, methyl groups. All of these, of course, if you don't have vitamins, if you don't have them, you can have certain deficiencies that show up, and so this is why it's important to know that you're getting, I guess, the right amount of vitamins, or you could always correlate to whatever symptoms you're having and maybe figure out if you're deficient somewhere. Methyl groups and hydrogens with cobalamin or B12. Sorbic acid, vitamin C. This one fends off scurvy. Important for electron transfer. Biotin, vitamin H, carbon dioxide. Something to do with carbon dioxides. Okay, so like I said, I'm not going to ask you for most of or for really anything that has to do with the vitamins, as far as their specific functions. Know the vitamins by classification, so water soluble or fat soluble, but beyond that, you don't really need to know anything for the exam. Identify each of these as a simple enzyme or one that requires a cofactor. So, requiring magnesium 2+, that's going to be requiring a cofactor, right? Requires vitamin B3 to transfer an acetyl group. It's not going to be a simple enzyme if it needs something. Is active with 4 polypeptide subunits. So that would be a simple enzyme. Now, as far as our fat-soluble enzymes go, there's only vitamins, sorry. There's only a few, A, D, E, and K. These are not involved as coenzymes in catalytic reactions. And that's probably because they're soluble in fat and not in the aqueous solution where most of these reactions happen. They are stored in the body, so we don't have to get these from our food every day. But they are still important because they all have roles in our body. So from vision, bone formation, antioxidant function, blood clotting, they're all very important. Let's go through them. So vitamin A is an antioxidant. It's needed for retinol, so this molecule is part of the retinol reactions that allows you to see. Necessary to avoid night blindness, immune system repression, and slowed growth. So there's quite a few functions there. I'm only familiar mostly with the molecules' involvement in vision. We got vitamin D. This one we can make from sunlight if you are exposed to skin. If you expose sunlight to your skin. It regulates absorption of phosphorus and calcium, so again, important. Necessary to avoid weakened bones. We actually fortify milk with vitamin D because historically children working in the mines weren't getting enough sunlight. So, why get them out of the mines when we can just give them vitamin D in their milk? So that's vitamin D. Vitamin E is also an antioxidant, meaning it's there to help scoop up free radicals or any kind of oxidative damage that can happen as a result of just having oxidants inside your cells. So it's kind of a sacrificial molecule, necessary to avoid hemolysis and anemia, so related to blood clotting. And then vitamin K also has a role in. Oh man, where's vitamin K? I guess I forgot to put vitamin K in here. I'm sorry. I don't have to draw it for you. Do I have to draw it for you? You guys don't care. Vitamin K kind of looks like. Got a couple of these units. You can have actually a lot of these little units on them. These little isoprenes. So N just means multiple. We can have 1, 2, 3, 4. So these guys, vitamin K, these are needed for the synthesis of zymogens for blood clotting. So responsible for making some of those inactive enzymes, and necessary to avoid prolonged bleeding time and bruising. So vitamin K, otherwise called menaquinone. So those are the fat-soluble vitamins. So here, knowing fat-soluble versus water-soluble is really the only objective. So if you look up folic acid, you'll remember that's a water-soluble. Retinol, we just talked about, fat-soluble. Vitamin E, fat-soluble. All of those other ones, water-soluble. Really, the fat-solubles are the short list, right? A, E, K, and D. Sorry. Another learning check. Identify the vitamin associated with each of the following. Again, I'm not going to ask you any of this stuff, but this is nice to know. We are going to move on and talk about FAD and NAD and some of these a little later in metabolism. But as far as this part goes, this is just seeing if you guys can, kind of look up some of these. So collagen formation is ascorbic acid. Part of the coenzyme FAD and FMN, that's riboflavin. Absorption of phosphorus and calcium in bone, we kind of talked about that. Vitamin D, vision, vitamin A, blood clotting, vitamin K. Sort of the what is the word? Sort of the main kind of goal that they do, right? Or the main job that they do. They do other things as well. All right. Oh, there it is. Somehow I got moved around. My bad. All right. Concept map. So this just shows you sort of what we've been talking about. Proteins that are enzymes. They have an active site. They interact with substrates. They form products. There are ways that we can regulate them, right? So regulation, of course, activity is affected by some of these conditions. And then we also talked about inhibiting, which isn't exactly shown here. And then we talked about cofactors. So don't forget, inhibitors were also talked about. Yeah, missing that here in my concept map. Sorry about that. So affected by, regulated by, and then inhibitors. All right, guys, that wraps up this chapter. It's a long one. Sorry about that. A little over an hour and 20 minutes. But let me know if you have questions. And, of course, use the discussion board or hit me up with email or on Canvas, and I can help you out. But hopefully this wasn't too bad of a chapter. Yeah, don't worry about the vitamin specific activities. Just kind of know where they fall classification-wise. All right, guys. See you in the next one. Take care.